So it's a great pleasure to be here. Thanks to the organizers, and thanks to all of you for coming here. So it's a particular pleasure to be coming back to Boston because I did a postdoc here at Harvard Neurobiology. And I was just starting my career, and neuroinformatics didn't exist back then. And I feel a little bit like Voltaire, who woke up one morning and realized that he'd been speaking prose all his life. And neuroinformatics now is something that is familiar, but it wasn't 30 years ago. It was a very different scene. Neuroscience was much more empirical, less quantitative, and that's changing very rapidly now. So the importance of what we're doing here is, I think, going to have a huge impact on the future of neuroscience and biology in general. So the focus of my talk is going to be on something a little bit different from what I've done in the past. And Kenji pointed out that I have projects at many different levels of investigation. I'm interested in the molecular organization of synapses. You'll be learning a little bit about that today, about how neurons are connected up in networks and how networks, the dynamics of networks, give rise to interesting transformations between sensory maps. And then finally, trying to understand how the entire central nervous system creates behavior. And ultimately, that's the goal, is to link together these levels. Now, what I'm going to be focusing on today is something that is outside the diagram, and most people ignore, and that is the space between all of these elements, the space between the synapses, between the neurons, between the glial cells, the extracellular space, which has not been explored, in part because it's very difficult to probe the extracellular space experimentally. We don't have any way of being able to even, for example, measure the space in vivo. What's the spacing between neurons in vivo? We can look at it in electron microscope, but that's after a lot of processing has taken place. And we know that it changes the relationships between the neurons and all of the other elements. But we can try to understand in a reconstruction, we can try to, as you'll see, try to compensate for that. Well, whenever you think of a neuron, what do you think of? Well, we have in our brains iconic images, and these iconic images have enormous impact on how we think about how the brain works. And in my mind, the iconic images that have the most impact are ones that were originally drawn by Ramon y Cajal over 100 years ago, great Spanish neuroanatomist, who perfected the Golgi technique, and with it cataloged the entire complement of neurons in the brain, which are still being mined today. In terms of neuroinformatics, he might be the patron saint. Now, how does this technique work? Well, miraculously, it's a reduced silver stain, which means that it's like a photographic picture, and miraculously, it only stains 1 out of 1,000 neurons, but when it does that in its entirety. Not complete, doesn't do the axon as well as it does the dendrites, but nonetheless, Cajal is able to produce magnificent pictures that have never been seen before. Never imagined, because of the fact that the great ramifications here and the detail, the spines on the dendrites, haven't really been seen in their full glory. Now, this is not a photograph. This is a drawing. I'm going to emphasize that he used to go through his microscopic slides, and if you've ever gone through a Golgi, it's not quite as clean, right? There's typically a lot of cells that are kind of mooshed up, and you only have a section, you may not have the whole neuron. So what he did was look through hundreds of slides, and then at night he would draw a composite of all of the neurons that he saw, and this is kind of like a prototypical pyramidal cell. But he got all the details right. There's the axon is branching off, the apical dendrites, the basal dendrites. It really, it's just amazing. Not only that, but he also described in great detail many of the things now that many of us are studying. For example, the sizes and shapes of the spines coming off of the apical dendrites. He describes here the density. For example, there's about 1 every micron on these particular apical dendrites. And he also described the circuitry in different parts of the brain. The retina, he basically set the pace by describing dozens of different types of amacrine cells, many different ganglion cells that were described that were different because they had different dendritic stratifications in the inner plexiform layer. So he was able to show that even back then that there had to be multiplicity of different channels that were going through the retina that were sending their axons into the brain, and it was long before we were able to understand what the different messages meant that were going there. But we now know there's like 20 different channels, all carrying different types of information, but in parallel into different parts of the brain. Now, hippocampus is particularly interesting. Of course, it's the structure that we know and love from the perspective of memory for events and specific items. It's part of the brain that is very well studied by electrophysiologists. They love it because of the fact that it's a very clean separation here between inputs and outputs. Cajal described not just the anatomical structure, but you can see the arrows here, which was his intuition, because there's obviously these are static pictures, intuition about the flow of information. He somehow deduced that the information was flowing in from the peripheral pathway into the dentate gyrus on these granule cells, which then flowed into the CA3 region here onto these pyramidal cells that had recurrent connections, and then from CA3 down to CA1, Schaffer collaterals onto the CA1 pyramidal cells, and then out here. And so that the whole trisynaptic arc was something that he really had in a sense, figured out just by looking at images. In fact, this is a model. If you think about what a model is, it's trying to infer function from structure. Now, what we're going to be focusing on for the next half hour is a little tiny bit of the neuropil, which is outlined here. It's the CA1 neuropil, and it contains many synapses from the Schaffer collaterals onto the dendrites of CA1 pyramidal cells. And as I mentioned, this is, these synapses are ones that have been extremely well studied by electrophysiologists who are interested in long-term potentiation and long-term depression. So a lot of the mechanisms that are maybe important for long-term memory may be contained in that little volume. If we could fully understand down to the last molecule exactly how those interactions are taking place. Now here's a different picture of what the brain looks like, right? So the cow picture is kind of this very idealistic, pristine picture of these neurons sitting there in the middle of, in a vacuum. That's not what it's like at all. When the electron microscope was invented and we saw the first pictures in the 50s, we saw something that looked like this, which is incredibly intricate and complex and very difficult to even think about because it's a two-dimensional cross-section. We're anatomists, of course, like Mark Ellisman, have devoted their careers of understanding every little component that you see here. I'm going to point out just a few of them just to give you a roadmap if you aren't familiar. So this is the outline of an apical dendrite, the kind that you saw from Cajal. In terms of this is a 1 micron bar. It's a 1.5 microns in diameter. It's characterized by microtubules, mitochondria, a lot of ultrastructure here that can be used to pick out what this may be, what element it might be. This is started with synapses. I'm just going to point out one synapse. The presynaptic terminal specializations, this is the vesicles. It's about a 0.5 micron across. And then postsynaptic spine head here, you can see the postsynaptic density. Okay, well, this is all the parts that you're familiar with, but what about the extracellular space? Well, if you look at it carefully, it's really hard to find because of the fact that the cell bodies are budding all of the synapses, and the synapses are budding glial cells that you can't really pick out very easily. So if you look at it, in fact, in the EM, it's actually less than about 10 microns, 10 nanometers. It's very, you can blow it up and see it's there, but in the textbooks it'll tell you that it's really more like 20 nanometers. And one of the reasons for that difference is that there is a process dehydration during which you have to suck away the water, and it's thought that that collapses the membranes down. So, but the problem is we really don't know, because we can't freeze the membranes fast enough if you really like to stop it in mid-flight and be able to see what it's really like in vivo. In fact, it's not probably fixed. In vivo, it's probably changing, but we don't have the techniques to follow those changes. Well, the next best thing is to try to reconstruct that neuropil and then try to bring it back to life, and that's what I'm going to be telling you about. So this is a project in collaboration with Kristen Harris. Kristen, for many years, has been reconstructing different parts of neurons, dendrites, spines, and so forth, usually one at a time. So we decided to team up with her and do the whole thing, just to reconstruct an entire chunk of neuropil. And the chunk that you saw there was about 5 microns on the side. So the process consisted of 2 different stages. The first is segmentation, which is very difficult, even for humans, looking at these electron microscopes. They're not nearly as crisp as you'd hope. And there are groups today that are trying to automate that. But so far, the gold standard is the human sitting there and drawing outlines. And that's what Kristen did. She provided us with a stack of 100 serial EM, transmission EM cross-sections, spaced about 45 to 50 nanometers, with roughly 5 nanometers resolution in plane, and each one of the processes is outlined as for example, this one here from plane to plane. Problem is, it's a big jump from plane to plane, so the problem of trying to link together the corresponding points, there's missing information, and so it's not completely clear how you connect these. Justin Kinney, who's now a postdoc with Sebastian Sion, in my lab, took upon himself the task of doing that for every single element. And he did it with collaboration with computer scientists who are experts on computational geometry. So this problem is one that's not specific to neuroscience. It's found in many, many areas. And there are algorithms out there that you can use. So Chandra Bajaj, in particular, who's a colleague of Kristen, helped us to follow every single process, axon, dendrite, and astrocyte, and this is what you end up with. It's like one of these puzzles, Chinese cube. And it's color-coded, and you'll be seeing more of what it looks like, but you can see it's as opaque as the actual tissue in the sense that all little pieces are intricately connected together in some three-dimensional structure. Well, how can we visualize that? So let's try to imagine what it's going to be like, and it's really amazing how different you think about a problem when you see it in a different way. And so I'm going to be showing you now a movie that was put together by Justin, which is going to try to illustrate all the different pieces and how they're connected and how they're related to each other. So here's a central apical dendroid going through that entire 5 micron cube. There are 77 spines. And at 5 microns, that means 15 microns per linear micron. These are in red, the active zones, the postsynaptic densities. And they come in a great variety of sizes and shapes. Here come the 75 axons that are connected to all those synapses, just the ones on that one dendrite. Volume, there is an astrocyte. It's a single astrocyte, and in fact, there's only a small part of the astrocyte which extends for 30 microns. And it's a continuous structure. And it fits in there like fingers in a glove. Okay, so this is only a bit, you wouldn't say anything if all the pieces were there, but before we put the Chinese puzzle together, let's dive down into the apical dendrite and see what's inside. And we see the endoplasmic reticulum, which is continuous and only shown here in like fragmented pieces because it wasn't fully reconstructed. But running down the entire length of 5 microns is the snake, which is a mitochondria. Not like the little jelly beans that you often see in the textbooks. Now we're going to find one of the spines. This is the spine neck, which is about a micron long. It's like a little tunnel. And we're going to spelunk through the tunnel and down into the head of the spine. And in red is the postsynaptic density. We're going to pop through that and look back. And now you can see the limbs, the neck and the spine. Now, one of the things that you read in textbooks is that the glial cell completely surrounds the synapse, and the reason is that it's the only way that glutamate is taken up, or the primary way is taken up. There are some pumps, some transporters on neurons, but the bulk of it is done through the astrocyte. And here is a case where, in fact, the astrocyte abuts the synapse. But in fact, the majority of the cases, the astrocyte is nowhere to be seen. But in spicules, whose function I don't think anybody knows except maybe Mark Ellisman. So here's the classic synapse, and this is the way that you think it should be. Here's the astrocyte completely surrounding the presynaptic and the postsynaptic elements, sitting there waiting to take up the glutamate. But by far the majority of the synapses, as you'll see, are pretty far away from any glial, within a few hundred nanometers. Okay, well, now let's reassemble, bring everyone else to the party. This is called the waltz of the dendrites. So these are all the other dendrites. There are 150 of them in that one little cubic, roughly 350 cubic micron chunk of neuropil. Here come the roughly 400 axons, filling in all the holes. At the end, there will be no holes. And now, the postsynaptic densities have been indicated in red. We just dissolve the dendrites so you can see what is left. It's like a Swiss cheese. Central dendrite. Okay, now here's the astrocyte by itself. And now this is something that would be very difficult to reconstruct in your mind if you're just looking at EM cross-sections. These are the postsynaptic densities of all the synapses. And you can see what the relationship is, right? It seems to be pretty much random. And most of them are floating in the middle of nowhere. But nonetheless, the glutamate is taken up. And we know that because we actually did the simulations. And finally, we'll end up with where we started. This is the central dendrite and the neuron within the neuron, the intricate compartments within the dendrite itself. Okay, well, just seeing that gives you a different impression. It really makes the problem, creates problems, actually. But there's a gold mine of information. I'm going to just that you can dig out from this, and I'm going to give you a taster of the projects that we're working on, just to see the different directions you can go. So one of the things that you noticed was the fact that the spines come in a variety of sizes and shapes. And here are the 77 spines in that one central dendrite that you saw. And you can see that some of them have huge heads. Some of them have tiny little heads. Some of them have big, long stalks, and some of them have practically nothing. There's no correlation whatsoever between the neck length and the spine head size. It's likely to find a big head on a small stalk as on a long stalk, right? So that's interesting, because it means that whatever function the spine has, it's it doesn't matter how far away you are from the dendrite. However, it turns out that there is some very, very strong correlations, and in particular, let's look at the correlation between the spine head area and the spine head volume. Now, if it was a perfect sphere, it would be a 2/3 power law, right? Area over volume. It's not, but it follows another law, which is a 3/4 power. Why? Who knows? But it follows it very accurately. It's telling us something about the shape. It's like a spheroidal type of shape, flattened sphere. Now, but even more intriguing is the fact that the postsynaptic density, we can measure that very accurately too. That also scales with the volume of the spine head, but it doesn't follow either the 2/3 rule for the sphere or the 3/4 power for the actual area. It doesn't scale with the area. Instead, it scales linearly with volume. Why is that? Well, maybe it has something to do with the concentration. The concentration has to do with the volume, the number of calcium ions. And maybe the PSD has something to do with the number of calcium ions coming in. Maybe it's important to match those. But it's probably not an accident that the two are matched. Let me give you another example. And this is something that we were still puzzling about, because there's a real mystery here. So spine heads come in tremendous variety of sizes or magnitude, right? And we think that somehow the spine head volume, since it's proportional to the postsynaptic density, is probably proportional to the strength of the synapse, right? It makes a lot of sense, and there's a lot of evidence for that. Chuck Stevens has done analysis where he's looked at the strength of the synapse, the probability of release, and then reconstructed the size of the spine, and it turns out they're related. But we noticed that unlike the textbook that tells you that the Schaffer collaterals go through perpendicularly and make one synapse at most with each dendrite, that there are several in the reconstructed volume. And as you can see, one here, here's the axon going through the volume, making a synapse here onto that apical dendrite, and then a second synapse on the other side. So there are pairs of synapses, right, onto the same shaft. Well, we asked the question, what's the distribution of sizes? If you have a big one here, is it likely to be small or big here? And so we went through our reconstruction, and we found there were 15 pairs on the same dendrite. And what you're seeing here is the distribution of the differences in volumes, right? Now, if the distribution was the same as just picking random spine heads, in fact, what we did is we picked 19 pairs where a single axon made two synapses on two different dendrites, right? This is what the distribution would look like. But you can see the differences are clumped up here near 0, which means that of the 15, there are actually 12 that are indistinguishably the same volume within the measurement error. Now just think about that, right? So how do these synapses change their volume? We think it's through experience, we think through presynaptic and postsynaptic activity, through time-dependent plasticity. Whatever the mechanism, it has to be incredibly accurate in order to be able to reproduce the volume, right? Now, in fact, because it has on the same shaft, it has probably the same postsynaptic history in terms of membrane potential. On the presynaptic side, it has the same axon, so it has the same action potential distribution. However, we know the release is probabilistic, so there's a stochastic element here, but nonetheless, somehow the machinery that regulates the size of the spine was able to match the two. And it might be telling us that despite the broad distribution of sizes that we see there, that it's not random, that in fact the sizes have been very tightly regulated, and that the mechanism for plasticity is also very precise. We don't know that for sure, but this is evidence. And the fact that it showed up in such very statistically significant, 0.003, means, on a small sample like this, it means it must be extremely significant in terms of the large scale cortex. Just think about it from the perspective, you just pick out a small little subsample and you see it. It must be the case that it's it's much more widely distributed. Okay, so now let's turn to the extracellular space. So the first thing that we did was to rescale it, and we did that by just linearly increasing all the dimensions by 15%. Roughly estimating, what the shrinkage is. And then we did some analysis, and one of the things we did was to ask the question at every point on every piece of membrane in the neuropil. We construct a little cone, and we ask what's the minimum distance to the next point. We use that as our estimate for the spacing, extracellular spacing. Now, in some cases, the cone hits more than one process. That occurs when you have 3 cells or more coming together. And we label those blue, and we're going to call them tunnels because in 3 dimensions they form these cylinders. And we also measure the distances in that tunnel. And then here's a cross-section with the 2 different designations colored in red for what we call sheets, surfaces between 2 cells, and then tunnels between 3 or more. And this is a purely topological definition, right? It's just what I just told you. So this is all now something that can be automated. Okay, well, normally, we think of the extracellular space as the outside. Well, let's think of it as the inside. What if it was a volume of its own? What would it look like? And here's what it looks like. It's this incredibly kind of velate, astrocyte-looking surface. And in fact, what's missing, of course, are all of the somas and the axons running through it and so forth. But this is what the topology looks like. This is what the space that the ions have to diffuse through looks like. And now, superimposed on that, what we did was to look at every single point, and we thresholded the distance to the next membrane, right? Remember finding the minimum and then looking at that number. And if it was less than 40 nanometers, it's colored blue, and if it's greater than 40 nanometers, it's colored yellow. And you can see immediately that there's 2 distributions here. There's one on the surfaces, which is between pairs of cells, say two flat surfaces, and then there are these the corners here we call the tunnels, because if you join them up, you can see they form a network of linear features that course through the entire extracellular space along these ridges here. So that's an interesting dichotomy, and but now we want to try to make this more quantitative. So there are a few things that we need to match. One of them is the extracellular volume. So it's known that the total volume of extracellular space is about 20%, 0.2. And if you look at the raw reconstructed EM and ask what's the total fraction of extracellular space there, it's below 10%. So that is an indication that we have to do something more than just expand by 15%. And so what we did was our first pass, our first approximation, is just put springs between all the corresponding points and just let the whole thing relax into a new configuration. So the idea is to create not just the original reconstruction from EM, but a whole series of reconstructions with different spacings that we could then compare and calibrate to the measurements. And indeed, so that's what we did. Here's the width, and for each one of these models, each one with different average spacing, we calculated the extracellular volume, and what you can see is that in order to reproduce the 20% volume fraction, you need to have a width of about 40 nanometers, which is about twice what you see in the textbooks. Textbooks tell you about 20 nanometers. But we know this is really crude because we expanded everything equally, right? It's probably not the case. So, but nonetheless, it tells us that we have to really be careful about trusting the textbooks. So what Justin did is to, as I create a whole series of reconstructions. Each one of them preserved the extracellular volume fraction, 20%. And what he varied was the fraction in the sheets and the tunnels. So first of all, let me just orient you. Okay, this is taking a raw EM and calculating the distances the same way we did in the reconstruction. And you can see that the sheets tend to be smaller than the tunnels. There's a big distribution here, but the median is farther away. And the same thing is true in our reconstruction. So we took that, and here's a series of 3 before expansion by 15% after expansion. And here in this pair, we try to create the medians of the sheets and tunnels to be the same, but still getting a asymmetric expansion here on the right. And then each successive one then had a higher ratio of tunnel area to sheet area. And you can see the median sheet distance goes from 40 nanometers, which is what we did in our first, this is our first pass, from 40 to 20 to 10, right? And you can see that in each case, the tunnel, median tunnel distance is going up at the same time in order to preserve all the relationships to get 20%. So this is, what we think is that reality is somewhere in between here, right? These are the 2 extremes, and it's probably somewhere in between. And if you had a bet, okay, 20 nanometers for the spacing between the sheets, and then if that was true, this would tell you that the spacing in the tunnels is huge. It's on the order of 80 nanometers. By the way, this is consistent with another experimental fact, which is that you can get diffusion of quantum dots through the neuropil, and these are like 60 to 80 nanometers in diameter, and no one could explain that based on a 20 nanometer spacing in external space, right? But it makes a lot of sense if you have a whole highway going through the neuropil that is much bigger in diameter. Just to show you that you can do a lot of quantitative analysis, I'm not going to go through this in detail, except to say that the median tunnel extracellular width is anti-correlated with the meat sheet. You saw that already. I showed that. We can compute by in our model by using Monte Carlo techniques to follow diffusion of molecules. We let go 10,000 molecules and see how far they go, and compare that with what would be in free space, and we can get a sense of the tortuosity. In other words, how much is the geometrical distances different because of the intricate connections between all of the different elements. And you can see that for these, there's 6 different models. They follow a nice regular relationship here, which is quadratic. And the measured tortuosity is 1.45, so this includes not just the geometry, but also the fact that there's extracellular matrix and things that are impeding the molecules from flowing as if they were free space in free water. And the ratio, the range here, as you can see, is constrained between 1.2 and 1.4. Somewhere in between here is probably the true value of the geometric tortuosity. And then putting together the total tortuosity and geometric, you can compute what the diffusion constant is, and it turns out to be somewhere between probably somewhere in the middle here, probably around 0.7, which is actually a lot higher than it is from previous estimates. So we can come up with really good quantitative numbers here, I think, that can really be used to pin down those values. But what we end up with at the end of the day is something really, really interesting. It's a meshwork, and we know that the diffusion is actually going to be faster along the tunnels, something we can measure. But the volume actually within the sheets is less, and that means that for a given number of molecules, you'll have a higher concentration. Of course, that's where the synapses are located on these flat sheets. This is the synaptic cleft. Interestingly, there's more tunnels around the synapse. Right, so you have a flat sheet, and then you have tunnels around it. And all these things, of course, could be exploited by nature in order to be able to adjust the signaling and transport of molecules. We know that there's volume conduction occurring in the neuropil. Okay, now let me quickly, so now what time do we have? 5 minutes left? Okay. Focus on what's happening inside of the spine. And here we use MCell, which is a simulated node. You're familiar with it, which tries to track every single molecule as it is released from the presynaptic terminal, diffuses over, binds to the receptors. We use the kinetics that have been measured regarding the rate of binding, opening and closing of channels, entry of calcium into the postsynaptic spine. And the idea here is to be as accurate as possible given the experimental numbers that the experimentalists give us, biochemists, from the rate constants, and the geometry from the anatomists. So here's a few snapshots in the simulation, and you can see that the release of 2,000 glutamate molecules spreads out very quickly, 15 microseconds, and then shortly thereafter, over a period of 100 milliseconds, we get entry of calcium, which are these little white dots. And just to animate that for you, here's a single release of a single, it's a mini of a single. There it goes, it's fast, and the postsynaptic receptors here, they show red when it binds to the NMDA receptor. Of the response, typically there's only a few receptors that are open. So we're talking about handfuls of molecules in terms of neurotransmitter molecules, receptors, opening and closing. It's very stochastic. Now, another thing that has been very well studied is spike time-dependent plasticity, which was originally discovered in the cerebral cortex by Markram and Salkman here in the hippocampus, B and Prue, showing that if the delay between the presynaptic and the postsynaptic, or in this case the presynaptic and postsynaptic, is within 10 milliseconds or so, if you pair that at about 20, 30 hertz, you can get a very nice doubling of the synaptic strength, but within that narrow window. Here's two pyramidal cells showing you the number of connections between them. So it's not a single synapse, it's probably like 5 or 10. And if you have the postsynaptic spike first and then the presynaptic and you pair that, you get a depression. So this is very well established. Well, let's try to simulate that to find out what's happening inside of the postsynaptic spine. And if we just have a single release, this is what you saw. You have an NMDA receptor, a few of them opening. And of course, the postsynaptic membrane potential is low, and you need to depolarize in order to open up more of the NMDA receptors. With the backpropagating action potential, you open calcium channels, and that's shown here. So here's the calcium input. The calcium is around, with calcium binding proteins, is actually a few micromolar. But if you pair the two, right, so if you have the EPSP within 10 milliseconds in here, delay of the backpropagating action potential, you get a much more activity in terms of the opening of the NMDA receptors and much more calcium coming in, up to 10 micromolar. So what this is telling us is that we can really titrate the entry of calcium depending on these events, pre- and post-synaptic events. And so this is the first step. Entry of calcium, we know, it just triggers the long-term potentiation. And we're collaborating with Mary Kennedy. Here's a paper that she published in PLOS on CAM kinase 2. Calcium binds to calcium, the calmodulin. Calmodulin binds to CAM kinase 2, and there are dozens of sites where phosphorylation takes place on the subunits. Right, there are a number of subunits. And the whole structure is very interesting because at a certain point it autophosphorylates. So the total number of possible states is like 10 to the 10th. You couldn't possibly keep track of every single one of them. But what we do in MCell is just keep track of the ones that are actually instantiated at any given time, and then make the transitions and only keep track of the state of the molecule itself. And this greatly simplifies the simulation. And so Mary Kennedy's group now is then integrating what she's done with our model, and we hope to be able to march down the pathway toward the mechanism, the actual induction of long-term potentiation. Okay, the last thing I want to tell you about is the fact that go back to the issue of the spine neck length. So just focus here on the bottom. We simulated in many different size in fact, all the variety you saw of spine heads and neck length and so forth, and asked what's the peak calcium. And so it turns out that the neck length has nothing whatsoever to do with the peak calcium levels. Now that's interesting. It's uncorrelated with the size of the head, it's uncorrelated with the size of the calcium peak, what's going on. And it turns out that it's because of the calcium pumps in the head. And so here's a normal condition, normal density of calcium pumps. And we can do something in a simulation that would be very difficult to experimentally. We tag every single calcium molecule, and we ask, where does it go? It enters the head. Does it go out through the neck? Does it go out through a pump? Does it hit a calcium-binding protein? Ultimately, it's got to escape somehow. And so here's, as a function of time, the ratio changes. Initially, the calcium-binding proteins have very high affinity. They suck it up, but then they eventually have to give it up. And then it is extruded over to the neck of the pump, and you can see that 80% or on the order of 80% are extruded through the pump, and only 20% through the neck under normal conditions. However, if we reduce, to really prove this is the pump that's important, if you reduce the density down to 1%, now we have over 70% going out through the neck. So we think that one of the reasons why the head, the calcium handling in the head is merely independent of the dendritic shaft is the fact that it's all being regulated internally, that the idea is that the purpose of the neck is simply to be able to have a connection with an axon, maybe a micron or two away, and not have and make the calculations in terms of the induction of plasticity and everything completely independent of what's happening in the dendritic shaft, except for the membrane potential, which of course has no trouble going through the neck. Okay, so I want to end with another movie, and this one is going to try to give you a sense for what it would be like if you were in the neuropil when of a rat, by the way, when you're asleep, you're the rat, and there's a sharp wave coming from CA3. This is something that Neurobiology has discovered. There's 100 millisecond bursts of activity spontaneously generated in the CA3, independent of any input from the cortex. In fact, it originates in CA3, then bombards CA1 with a firestorm of synaptic activity in the net that is transmitted from CA1 back to entorhinal cortex, and then from there back in feedback connections to the rest of the cortex. So what would it be like, we asked, if you simulated that barrage of action potentials, what would the synapses look like? And so that's what we've illustrated here. And the time scale is logarithmic. So it's going to start out on a very fast time scale in microseconds. And you'll see what it looks like for a single release. And then as we pull back, we'll see what the entire field of our volume looks like. Okay, so glutamate in white. Now, I wonder if we can turn the volume up. Okay, when it binds to a pump on a glial cell, it turns yellow, and if it's irreversible, it turns red. So you can see this is a single, and you can see the extracellular space actually very nicely here outlined. That's a single release. So here's another release. And you can see now as the time scale speeds up, the pumps are working away, and the dying embers represent the end point in a single release. just want to say that, in addition to Justin, we have many other researchers working on these projects listed here on the side, and collaborators, Mary Kennedy and Kristen Harris, I've already mentioned them, and also computer scientist Sandra Bajaj. I also want to say that this really, this method, there's two different ways you can do these simulations. One is, finite elements, but the trouble there is that you get these little volumes that are, there are no calcium ions, and so what's the concentration if one calcium ion comes in? So that's why we went to the Monte Carlo techniques, and really that goes back to Ed Salpeter. Tom Bartol, who was one of the architects of MCell, was a graduate student of Miriam Salpeter's lab who studied the neuromuscular junction, and she wanted to come up with a quantitative model. She asked her husband, who's an eminent distinguished astrophysicist, what to do. He told her that physicists would apply Monte Carlo techniques, and that's what they did. And then so Joel Stile, who was at the time a postdoc, and Tom got together. This is well over 20 years ago. And the first MCell simulator, before it was called MCell, was written in Fortran. Since then, computers have gotten thousands and hundreds of thousands of times faster. It's made it possible to reconstruct and simulate all of the neurotransmitter release and all of the calcium coming in that little volume of neuropil. And of course, with the memory that we now have, we can do even larger reconstructions. And I call this microconnectome because it's a micron scale, right? And it's amazing how much you can get out from doing that as opposed to the macroconnectomics, which a lot of other people are working on, which has more to do with reconstructing cubic millimeters of tissue and looking at the overall patterns of connectivity. Here we're focusing on the actual signaling going on within a small volume. Well, thank you very much for your attention.